गुड
नम
आफ्टरनून सर।
गुड आफ्टरनून। नमस्ते राधा किशन जी। Okay, so we have 10 people joined. Um, do we have information on rest of the students joining? Okay. It's 3-6. Let us start. Unable to see my screen.
Yes, sir.
Okay. So today we are going to talk about drug design of inhibitors against dengue virus envelope. Component one and salbutamol. Before we go into drug design, as I told you the other day, we need to find out which particular part of the protein or which particular part of the uh, uh, target or organism or whatever it is that we are trying to design a drug against. We have to find out which is the most vulnerable part or which particular things can be targeted. And to know that, you need to identify the life cycle, or you need to identify what happens with the particular pathogen. So in this particular case, in 2008, this particular life cycle of the dengue virus it was taken from this particular slide. But the life cycle of dengue virus is not nothing new. And as you may know, every summer in India, we have a epidemic of dengue, and we have many people who have died because of dengue hemorrhagic fever. So, dengue fever and dengue hemorrhagic fever are two different things. So, let me just clarify what that means. So, dengue fever is the first instance you have the viral infection, and the virus creates a havoc in your immune system, because of which you have very high fever associated with joint pains, and it usually subsides within a week or so. There are no known treatments for dengue virus and viral fever, and the whatever treatment that they give in the hospital is platelet replenishment and symptomatic relief, meaning that they will give analgesic, which will reduce the body temperature. They will give painkiller to reduce the joint pain and so on. So in dengue virus, there are four serotypes. So if you are infected by serotype, let's say one, and then you are infected by, let's say, another serotype, what happens is there is a cross reactivity of the antibodies. And because, excuse me, and because of this cross reactivity, you end up getting a dengue hemorrhagic fever. Now, dengue fever fatality, if it is about 30-40%, when you get dengue hemorrhagic fever, because of the cross reactivity of the antibodies and the antigens, the fatality rate is very high. And usually, whatever you see on news in the month of summer, when in Delhi or any other cities where dengue fever is more prevalent, you will hear the people dying because of the dengue hemorrhagic fever and not actually dengue fever. But in India, we have serotype 2, which is more prevalent. And in other countries, you have other serotypes very prevalent. So when you have, but that doesn't mean we don't have other serotypes in India. We do have other serotypes in India. And the cross-reactivity, reinfection with another serotype makes the dengue hemorrhagic fever pretty fatal. Now, irrespective of the four serotypes, the dengue virus undergoes the life cycle in this particular manner, going from A to G in this anti-clockwise manner. So first, the virus, which is shown here, binds to the host receptors, and these host receptors are surface proteins that actually are signaling molecules, meaning that they can bind to something else as well as part of the normal process, and they will bind to the host receptor. And what happens is that once the host receptor is bound, the host receptor proteins, they will take the virus inside the cell. And this is where the endosome, this particular organelle which you see in yellow, is endosome. And in the endosome, the virus is now happily sitting inside. However, compared to the extracellular or the outer environment of the cell, where pH is mostly neutral, endosomes are in general very acidic in nature. So when it's acidic, sorry, do you hear any disturbance when I'm speaking? Are you able to hear like some mechanical sounds or something like that in the background? Can anyone confirm or say anything?
Very little.
It's clear, sir.
Very clear. Okay, then that's okay. There is some construction going on, and I was thinking if it is interfering. Anyway, so the endosome is an organelle that is present in any cell, any eukaryotic cell. And the reason it is present in the eukaryotic cell is to take uh, any particle that is not part of the host and degrades it. So that is the whole reason why the pH is very acidic to, because at a low pH, the proteins will denature. The function of the virus is completely different, and the virus cannot function very well. That's the usual general case. However, in the case of dengue virus, what happens is this low pH enables the virus to fuse. And what you see here is the fusion. So the viral proteins and the viral membrane, this is the RNA virus. It has a membrane around it, which is not shown here, but I'll show in the subsequent slides. So that viral membrane is also a lipid bilayer, and this endosome is also made up of a lipid bilayer. So those two lipid bilayers will fuse. And when it fuses, what happens is automatically now the viral content can be released into the host cell. So when the viral content is released, the viral protein will remain in the endosome. It will get degraded, but the RNA, that is the main part inside the virus, will release into the host cell. Now, the RNA, being a nucleic acid, what it does is that this is as per any virus that we know and how viruses behave. They hijack the cellular machinery of the host cell. So they take up the protein translation machinery of the host cell, and then they start producing the entire genome, multiple copies of this RNA genome. And at the same time, the RNA can be directly translated into the proteins, whatever proteins it's required, the structural proteins of the dengue virus. Now, once the RNA is translated and RNA is replicated, it will be attached to the membrane. And in the membrane, what happens is that it gets assembled into an immature virus in the endoplasmic reticulum. Now, endoplasmic reticulum's pH is slightly higher, around neutral. And what you see here is that it undergoes this, and then it forms the budding of the endoplasmic reticulum. And in the endoplasmic reticulum, all the viral assembly happens. These are immature virions, as we call it. And as we know, in any host cell, the endoplasmic reticulum and the Golgi body, they are related, they are connected. So through the Golgi body, the immature variant is transported. And during this process, it matures. And during that part of maturation, there is a protein that is cleaved. So that is called PRM, that is a precursor M protein. So that precursor M protein is cleaved by an enzyme called furin. By the time it reaches the extracellular part and gets released into the outer part of the cell, the pH gradually decreases back to a very low pH. What you see in the blue is the protein that is cleaved, and the proteins that are not cleaved, which are part of the still the virus, they are the mature part of the virus, and the virus cell cycle again goes back and then continues multiple times. During this entire process, what happens is that there are a lot of cells that die. The cells start becoming virus factories, and most often in the when it gets circulated in the blood, the platelet count gets reduced drastically. And that is one of the symptoms that is a diagnostic tool to identify if the uh, dengue viral infection has happened or is it is some other viral infection in the host cell. So this is the life cycle. Now, as I told you an analogy in some of the previous classes, the drug target you should think about, or what is the drug target in an organism, that should be thought about something like how an person assassinates someone. What do they do? They look up the entire day-to-day routine of that person. They check what time they wake up, what time they go to sleep, what is their routine, what time they are alone, what times they are most vulnerable when security is less, and that is the time that they go on attack. Right. So that's the entire process that we are using in one way to find out what are the loopholes in the life cycle of a organism or a pathogenic bacteria, virus, protozoa, whatever the organism is, and find out where they can be attacked. So in the case of dengue virus, there are multiple places where you can find out where they can be attacked. One is where they can be prevented from entering inside the host cell, or they can be prevented from replication of the virus inside the host cell. The RNA polymerase can be inhibited, or the protein translation machinery can be inhibited, and so on. Or that can be that the furin cleavage, which is required for immature virus to mature virus conversion, that can be inhibited, so on. So there are a lot of places where the viruses can be attacked. Now, in this particular work that we did, we focused on the envelope protein. Now, the envelope protein, let me let me show you this first and then I'll go to the previous slide. So in the envelope protein, this is the viral, the brown or the yellow color, cream color region is the viral membrane. And these are the dengue virus belong to the family of flavivirus or flaviviridae. So in flaviviridae, they are RNA virus, they are single strand RNA viruses that are covered by a lipid bilayer or a membrane. So these are membrane bound virus, which actually have some proteins expressed on the membrane. And one such protein is the envelope protein. The envelope protein, when you see on the viral surface, it will be packed one beside the other as a dimer. So here you have blue, red, yellow, one chain, blue, red, yellow, another chain. So they are kind of present as a dimer. Once these envelope proteins get recognized by the host cell, they are taken up inside the host cell. This is outside, and now this is inside. This is the endosome. This is the outer cell membrane in A. In B, this is the endosome. Now, in the endosome, because the low pH, what happens is this particular envelope protein undergoes a transformation from a dimer to a trimer. And two such trimers, what happens is it attaches itself or anchors itself to the host cell membrane in the endosome. And so it's kind of like a Velcro. And what happens is it starts pulling the two trimers towards each other, and it undergoes a huge conformational change. And then what happens is it establishes a pore. This pore now, what you can see, this is the end. So when it starts attaching, once it has attached, two such trimers will start pulling the endosome membrane with the viral membrane, and at some point of time they will start fusing, and once it fuses, it can create this pore. So this particular hypothesis of how the dimer to a trimer conversion happens, what are the factors that lead to its conversion, and also the viral protein, the envelope protein undergoes a huge conformational change, not in just the stoichiometry, that is a dimer to a trimer, but also in terms of conformational change in the structure. So what you see, this blue tube, which is the membrane bound region, slowly you can see that in the dimer it is not attached to it, but in the trimer they are bound with the protein itself. So we took up this particular entry of the viral protein as a target because this particular protein exists only in the virus, not in the host cell. And thereby, even if we attempt to um, inhibit this protein, any other host cell proteins will not be affected unless there is some cross reactivity of the drug with the host protein. Also, there is a particular attraction or particular strong case to prevent the virus entering into the host cell. So if we prevent the fusion of the virus, that is, if we lock the viral protein in this conformation and don't allow it to go to this conformation, that is from a dimer to a trimer, then we can prevent any further host pathogen interaction where the virus can interact and start going downstream. So in this particular protein, what you see again the same coloring, blue, red, and yellow, they are colored for each domain. So it is a three-domain protein. And at the end, this green color is the fusion peptide. This is the peptide that latches itself or attaches itself to the host membrane. TRM is part of the immature virus that when cleaved, you get the mature virus. So you see this cyan color TRM here. Once the immature virus also, which is in a heterodimer, but not exactly in the form we know about, when it matures, it forms this raft-shaped protein homodimer. And after it gets activated at low pH, it forms this uh, post-fusion trimer. And another main difference is that this viral fusion peptide, which is present in green here, this which was not exposed in the dimer, now it gets exposed in the trimer. So the conversion of, or at least the dengue viral inhibition is completely because of the conformational change and also the fusion peptide activation or the exposure. So when we compare the structures of the pre-fusion and the post-fusion, we see that there is this conformational change between domain 1 and domain 2. So domain 1 and domain 2, sorry, there's actually domain 3 and domain 1, and domain 2 is colored in yellow. Now, this particular domain 3 and domain 1, the in between them, you have a series of conformational changes happening. Now, when we look at it, it looks as if this particular blue region flips over the red region, and it forms this trimeric shape or the monomer of the trimeric. But in fact, this between the domain 1 and domain 2, that is this particular part here, actually the domain 2 is what undergoes a huge change if we make the domain uh, 2 and 3 static. So in the immature virus, you have a angle of -6. What is it compared with? It is compared to the soluble envelope protein. And in the mature virus, which is here, which is present in the pre-fusion form, it is at an angle of 21 degrees. And in the post-fusion form, which is here, it is at an angle of -16 degrees. So this hinge angle, which is hinge meaning it is just like the hinge that's present in your doors. That's what makes the door open, and the angle that's made actually determines how the protein conformation is different between the immature, mature, and the post-fusion form. So even though if we keep this domain 2 static, then you will think that the domain 1 undergoes a huge conformational change. But actually, this particular yellow domain is what undergoes a huge conformational change. Now, as I said, there are 4 serotypes of dengue virus, serotype 1, 2, 3. At this point of time, when we did the study, we had serotype 2 and serotype 3 structures available. And the sequence identity between the serotypes for this envelope protein is about 40%. And the sequence similarity is about 50 to 60%, which means that between serotypes, the structural similarity is very high, but the sequence similarity is not that high, although it is at 60%. Identity is very, very less. And then, since this dengue virus belongs to the family of flavivirus, there are other flavivirus which are also pathogenic to the humans. Like, for example, tick borne virus, encephalitis virus, TBEV, West Nile virus, which is WNV, and Japanese encephalitis virus, JNEV. Now, if you look at all these 3 different envelope proteins of those viruses, since they belong to the same Flaviviridae family, their sequence may not be very high in terms of identity and similarity, but you can see that the structure is highly similar across the various parts of the Flavivirus. So, uh, there are the whole idea is that to prevent the flaviviral from entering inside the host cell. And this is, as I said, there are multiple places in the viral life cycle that you can think about where you have to, you can, or you can identify drug targets, and you can design a drug against them. But in the case of flavivirus, one of the most common drug design approach that we have known from early studies is the non-structural protein NS3 or NS5, and those proteins that have some kind of a function like helicase, protease, and so on. So that the at that point of time when we started this, fusion inhibitors were attractive, but there was not much being done about it. And what you see here are other antivirals that have been developed for flavivirus, not necessarily for dengue virus, which actually have been tested, and there are a lot of other indirect methods by which you can find out the inhibition of the dengue virus. But none of these antivirals that are listed are available in the market, or they have gone through the final steps of being commercialized. There have been toxicity studies, there have been some kind of um, preparing them in large scale and so on. There are a lot of limitations. So, and when we look at the surface of the dengue viral envelope protein, especially the serotype 2, which was what we took up as the first structure for our study, there are two reasons to this. One, it has a very high resolution crystal structure. And the second one is that it is the only serotype for which, at that point of time, we had both the pre-fusion and the post-fusion form. So for many of them, only the pre-fusion form was known. The post-fusion form was not promising enough to be crystallized, and they had to go to other viruses to do the crystallization. Anyway, the first thing when the crystal structure was released, uh, Yorgo Moldis, he found that there is a particular place in the structure, especially when it becomes part of the time. So red, blue, red, yellow, up and green. Up to that is one chain, and similarly the next chain is here. In between these two chains, you can see a cavity, and in the crystallographic data, that was bound with a beta OG, or we call as BOG. It's a detergent, and it was small enough that it was able to bind to the particular cavity. So there were parallel work that was happening in the same lab, where they designed drugs to target this particular site. And since this is a structural protein, there are only so many theories of enzymology that can work with dengue virus. Oh, so the objective of the work was first identify something else in the protein that can be targeted, and use the uh, existing dengue viral envelope proteins, their available sequences, and so on, to validate those cavities. Just one second. Oh, sorry about. Okay. So the second step was, once we have validated the cavities with the envelope protein, we had to perform a virtual screening and design inhibitors against this particular protein. And then, to understand the mechanism of this envelope protein and the role it plays in the viral fusion, we also did molecular dynamic simulation. So those are the objectives. Now, how did we go about identifying a cavity? Now, what is a cavity? It is the same definition that your dentist will use, that it is a cavity is nothing but a depression or a place where something gets lodged and it cannot, it is kind of preventing the, or it has a function associated with it. Now, what you have to remember for viral proteins is that viral proteins have both structural and non-structural proteins. Structural proteins are those that are part of the viral capsid and part of the mature virus. And this is the one that you will see when the virus enters into the host cell. Non-structural proteins are enzymes, functional proteins that are required for the viral life cycle, like the NS1, NS2, NS3, and so on. Some are helicase, some is a protease, and so on. So everything has a almost an enzymatic function associated with it. Since we wanted to focus on the conformational change that is happening in the dengue viral protein, this envelope protein is not a functional protein but a structural protein. It enables the virus to form a stable capsid, and it does a pretty good job of enclosing everything inside the virus, and it also bounds or it also envelops itself with a viral RNA membrane. And when the host cell is encountering the virus, the first thing that it does is to latch itself to this particular envelope protein. Okay. So the task is not to identify a cavity just like how you would identify in an enzyme, but you want to identify a cavity that is functional in terms of the viral life cycle. Just one sec, give me 1 second here. Okay, sorry. Yeah, so what we did the approach was we took an approach of evolutionary information, conservation of residues, and the structural information to find out cavities that are exposed to the receptor facing site. So what we did was we took the serotype 2 of the dengue virus. This is the PDB ID of the pre-fusion. This is the post-fusion PDB ID. And we took the envelope protein and we gave it to Conserve. Conserve is a tool that can identify which particular residues on the solvent accessible side is highly conserved and which ones are not. Then at the same time, we did the comparison between the two structures by superposing them. And also we used two different uh, binding site or cavity identification algorithms. So it is a cavity identification tool that there is one called PASS, the other one is called CASP. Come here. CASP that and the pass is the method that uses geometry based identification, and it fills up any depression in the surface of the protein with solvent and solvent sized molecules. So basically spheres with different radii. So a water molecule has a radii of about 1.4 angstrom. So 1.4 angstrom radius spheres can be filled in the cavity. Wherever you have the solvent binding or the surface of the solvent or the sphere binding, you can find out if a particular place on the surface has too many water molecules that can be filled. So basically, it's like packing water molecules into an empty space. In contrast, CASP, this is another tool where we'll look at the identification of a cavity in terms of area, volume. So basically, it will look at surface area, it will look at surface volume, and then it will rank how many cavities are there which are highly depth. It will not take into consideration shallow cavities. So there will be overlap between the two different algorithms because both are cavity detection algorithms. And there might be cavities which are predicted by both. There will be cavities which are predicted only by parts. There will be cavities predicted only by CAST. Taking this into consideration, what we did was we came up with a selection criteria. Now, for any protein, there will be multiple cavities. There will be 20 cavities, 30 cavities, many different cavities. Okay. So in the criteria is now something that is not present in the existing literature. The criteria is something that we have to come up with. Again, you have to think like an assassination attempt. What will give rise to this particular protein to get inhibited? So the first criteria we used was cavity that is present in the dimer but disappearing in trimer. Means when we use these two tools to identify cavities in the pre-fusion and the post-fusion conformation, you want to have a cavity that is present in the dimer form, but in the trimer form, that cavity is no longer present. What function does this cavity will have? It will be involved in a conformational change and the trimer formation. Like for example, if you go back and I show you the beta OG binding region, this beta OG binding region is present in the dimer. Okay. But if you look at the trimer, this is where the beta OG is. This particular region is no longer absent. It's no longer present. Sorry. So you want to identify cavities like this. So beta OG site actually fits the selection criteria. However, this beta OG site is already declared or already been characterized. So we had to find other cavities that have the same criteria. The second criteria that we used was the cavity is present in the dimer, which is responsible for dimer association, meaning that cavity is something which, if you let us say, think of it like a river on either side of a valley, and if you construct a bridge, this bridge will connect between both the valleys on each banks of the river, and thereby it will act like a stapler between the two monomers and prevent the dimer to dissociate. So in other words, it will targeting this particular type of cavity will lead to prevention of dimer dissociation, meaning you're locking the uh, confirmation in the dimer position. So looking at all the different cavities that we found, there were 6 cavities that fit both the criteria that were common between both the tools, and they were present in the pre-fusion and post-fusion. And then we looked at which of these cavities are on the receptor facing side. So this is the membrane. You can see the curvature of the membrane bound protein. So this is the membrane lipid bilayer, and whatever you see on this This is the dimer. The dimer has a membrane bound side and a solvent bound side. Now, the membrane bound side is automatically when you see a slight curve in the protein. The existing extracellular side, you have residues that are glycosylated. These are carbohydrate moieties that are bound with the protein. So, since it's a dimer, the same position of the carbohydrate technically can be bound with the residue on this side, but you do not have those residues glycosylated. So, you can see other labels H3, H6, H9, H5, all these mesh like characters with small mesh in this are all different types of cavities, okay, that we are predicting. So when we looked at these structure and compared it with the structure, we found among the six, two cavities that fit the two criteria. The first criteria is cavity is present in the dimer, but not in the trimer. So that was the cavity H8, labeled as H8. Then the second criteria was cavity disappears in the trimer and it's no longer present in the and there is no corresponding active site in the dimer. So that was labeled as H19. So those were the cavities. Now, what are the other properties of these cavities? This is very close to the fusion peptide, which means that it is highly functional in nature. 60% of the residues in that active site are conserved 80 and above in terms of sequence. And the cavity is hydrophobic. It also has a charge distribution. It is solvent accessible. One of the other things was glycosylation. Now, if I go to the next slide and show you this in Zoom, this glycosylation, like the carbohydrate moieties in a protein, are very flexible in nature. When they are very flexible in nature, they can flop around. And what will happen is it will try to, at some point of time, it will try to make a interaction with the uh, protein itself. So there is a chance that this might block the cavity that was identified in the E protein. If it blocks, then the desired drug may not bind to this particular cavity or this particular place. So, what is the distance between these glycosylated residues and the center of mass of this cavity? It was by about 15 angstroms on 25 angstroms away. So, asparagine 69, asparagine 155, 153 are all the glycosylated residues. Volume is 298 angstrom cube and surface solvent accessible surface area is about 1190 angstrom square. Similarly, same thing. In the other cavity, it is present between domain 1 and 3, which is also a hinge where conformational change takes place. So if you go back, this is that particular hinge between 1 and 3. Okay. So here also you have good conservation. The cavity is hydrophobic, cavity has charge residues, and the glycosylated residue for that is a little bit far away. In both cases, at least 10 angstroms and more are far. Again, it has a good volume. It has a comparable solvent accessible surface area. So when we look at these two cavities, this is the black circles indicate where those cavities are present. It is present here and it is present here. That means in the dimer, since this is a homodimer, H19 can also be present with another name. So we looked at that and then we eliminated the name and then we kept only one. And anyway, when you do virtual screening, you focus only on one particular cavity at a time. Now, this is the conservation map of the residues in the serotype 2 compared to serotype 1, 2, and 4. And then what you see is the distribution of charge residues and hydrophobic residues with histidines colored separately. So it tells us that there is a good charge distribution and good hydrophobic content, and these two cavities are good candidates for doing drug design. When we look at the structure of other flavivirus and other serotypes of the dengue virus, like dengue viral serotype 3, uh, tick-borne encephalitis virus, West Nile virus, and Japanese encephalitis virus. This is the center of mass of those cavities in all the other proteins. And when we compare those structures, we see them all having pretty good structural superposition. And the distances between the center of mass of these cavities is not very far. That means this particular site, which we now label it as site 1, is a pretty good candidate which is present in the dimer but is absent in the trimer. So that is the criteria 1. It is near to the fusion peptide. It's involved in the conformational change and the trimer formation. So now we went ahead doing a virtual screening. So in this structure-based drug design, because we know the protein structure, we know the structure of the molecules that we want to dock. So this is a structure-based rational drug design. So up to this part we had covered, and once we took the one OKE, the pre-fusion form, there were these many molecules that we took from multiple sources. Then we filtered those molecules with a drug likeness index using the tool filter, where it uses not just Lipinski's rule of 5, it also looks at number of hydrogen bond acceptors, the country where they are going to be procured from, the country where they had been designed, how easy it is to design, and so on. So then we did virtual screening of these molecules in 3 different tools: Gold, FlexX, and Dock. And we did a post-docking analysis using X-score, which is a independent scoring function. And we inspected each of those top ranking lists visually, and then we identified about 7 potential inhibitors. So these 7 inhibitors, they look like this. Then we looked at how do these 7 ended up being selected. We see that it has a aromatic ring near the carbonyl region of the protein, and they are very common in all the 7. And there is also some fine membered ring which is extremely common in all of these. Now, once we go to the mechanist or medicinal chemist and ask them how many steps are involved in synthesizing each of these, because when we are trying to buy this from a company, the company might sell it as 5 gram, 10 gram, 100 gram, or 50 gram, and higher the amount, more expensive it becomes. That is okay. But if you have to do some kind of compression, then okay. Up to this point, any doubts, any questions? Okay. Continuing. Now, we had a collaborator who was a medicinal chemist, and we took these molecules and asked them uh, how many steps would be required to synthesize these molecules. And they said anything that can be synthesized within 20 steps or less than 20 is a good molecule to buy or to test, because eventually these are all commercial molecules. you cannot patent them. You have to modify them, and then the derivation or the chemically derived molecule is what can be patented. So if there are more than 20 steps, the yield at the end either will be of low quality, or the particular molecule may be too hard to synthesize. Okay. The database in which this was found was Chem Division. It's from the company called ChemDiv, and this is the IUPAC name. And what you can see is the original score that was got from Gold, which was a virtual screening tool, FlexX, and Doc. Now, automatically, you will see that Gold gives a fitness score, whereas FlexX and Doc give a binding energy. Energy is the in terms of kilocalorie per mole, but fitness in the Gold is in terms of a number where higher the number, more closer to 100, it's a better docking. Now, the second thing that you will notice is that even though FlexX and Dock give in kilocalorie per mole, they both are not very similar values. Here it is giving minus 9.44 kilocalorie per mole. Here it is giving minus 19.82 kilocalorie per mole. So which one is true? Which one is the correct binding energy? And also, how do you compare the binding energy of FlexX and Dock with Gold, which is a positive integer? And in this case, they are all negative values, or positive values with negative values. The way we can do it is you can use an independent scoring function to recalculate the binding energy. So that is what we use, which is called Xscore. And it also gives us in kilocalorie per mole. And the kilocalorie per mole here, you can see immediately it is comparable. Here it is minus 9, minus 7, minus 7, minus 8, minus 7, minus 6, minus 9, minus 7, minus 6, so on. So what makes this particular uh, uh, comparison easier is it is in the same scale. One. Then the next question comes is, how much of a difference between the X-score values should you consider them as completely different type of docking? The way we do is that if two values of X-score, if they are within the plus or minus 2.2 kilocalorie per mole cutoff, then they will be similar. Meaning, if let us say one molecule docked with X score binding energy value, after docking and reevaluation, you get a value of, let us say, minus 10 kilocalorie per mole. Then if you use gold, then you get minus 8 kilocalorie per mole. You use FlexX, you get minus 5 kilocalorie per mole. Now, minus 5, minus 8, minus 10. The difference, minus 8 and minus 10 are much closer in number because they are plus or minus 2.2 kilocalorie per mole than minus 5. That means there is a disagreement with different docking tools when it comes to the same molecule bound to the same place. So what we did was we took a consensus approach where anything that a molecule is ranked higher in gold is also ranked higher in FlexX, is also ranked higher in DOC, and their X-score values are within the cutoff of plus or minus 2.2 kilocalorie per. So when we put this kind of filtering, we got a very comparable results where the X-score values of these molecules, even though they were docked with three different tools and they probably have docked slightly different way in the protein structure, they still are comparable in terms of energy. Then the next step, obvious step, is to find out what residues they are binding to. Give me a minute here. So in a particular docking virtual screening, the molecule binds in the most comfortable position in the most lowest energy conformation. That is what we have been talking about, we have been seeing in the previous class. However, two molecules can bind in the similar binding energy, but may have different orientations, which we call as conformation or pose. And the orientation might be completely in agreement or not in agreement. One second. Okay, so the how do we know without looking at the molecule if two molecules are having similar orientation or conformation? The easiest way you can do is find out what are the hydrogen bonds it's forming, what are the close contacts, and how much of those close contacts are conserved in nature. Because this particular protein is not an enzyme, it is a structural protein. We take this particular criteria of interaction very strong because we do not know which residues are important for the viral protein function. Okay, so the what you see here is R1. Among the seven, we label them as R1 up to R7. R1 molecule forms three hydrogen bonds. There are 14 non-hydrogen bonds or hydrophobic interactions. Among these, it makes close contacts of 13 out of the 33 contacts. The percentage is about 39.4%. Now, what the number in the brackets indicate is the number of bonds. So here serine is making one bond. Here serine is making two bonds with another molecule. What you see automatically if you look row in the column is that there are some residues which are repeated across the seven molecules, which means this particular residue, serine 363, is most amenable for binding to the small molecule. And when you look at the percentage of close contacts and number of interacting residues, you see some patterns. But whether they are meaningful or not is system dependent. In this particular case, we found that they are pretty useful in finding out or validating that the computational method of these docking is kind of okay. So, since we are going to order these molecules, we don't want it involves money and we need to purchase molecules that are completely different in nature, in the chemical diversity. So why is this important? For example, let us say R4 and R5. Okay. You can see that they belong to the very closely related structures. The structural overlap is very, very similar. And uh, if I study one, that's more than enough to study the other, which means I don't have to purchase this molecule here, or I don't have to purchase this molecule here. So when we mapped the diversity of the seven molecules, we found that majority of them are in different clades in terms of structural similarity. Some like R1, R2, R4, R5 are highly similar to each other. Now, when we did following this, I will shown the results. We did a MTT assay. MTT's assay is to determine the cell cytotoxicity. Any small molecule, when we take it up inside the human body, it gets degraded or metabolized in the liver. So the liver cells are the first set of cells that will encounter these toxic molecules. And if there are some toxic molecules, they will lead to hepatotoxicity. So an easiest way to find out if the molecules are toxic or not is to test with a cell viability assay called MTT cytotoxicity assay. So it is an ELISA-based reader, and the cells that are alive after some point of time will not show a color change, but the cells that are died will show a color change. And then we found that most of these are good molecules. They don't cause cytotoxicity uh, in a very significant way. What you see here is inhibition of plaque formation. So this is the experimental method after MTT that was performed to find out whether which among these are really good molecules that can be taken to the next step. Um, the next step in this particular process is nothing but testing whether it can really prevent the dengue viral from entering inside the host cell. And for that, we do an assay called plaque inhibition assay. In plaque inhibition assay, what you do is you take an agar plate and then you put a layer of bacterial cells, okay, or the host cells, and then you make the virus incubate with the chemical molecule. So the chemicals or the small molecule will have to go and bind to wherever they need to bind in the E protein or the envelope protein. And they will, if they are liking each other, then they will be in a tight configuration or a tight complex for a long period of time. And after a short incubation period, the while it is incubating, the cell agar plate that has the mammalian cells will be overlaid with another agar layer, much more different composition of an agar that has the virus in it, okay, which is incubated. So what happens is that this virus in the first layer of the agar is movable. It can move because this agar is much more different in consistency, and the host cells are kind of permanently fixed on the agar. So that can't move a lot, but the virus cells can move and they will go and they will start inhibiting the host cell. Wherever the host cells are infected and the virus makes more virions and it matures out of the virus, out of the host cell at the end gets destroyed. So what happens is you will see some clear zones or clear area wherever the virus attack the host cell, because more viruses are produced and it will attack more neighboring cells and more neighboring cells, more neighboring cells. So you will have something, a clearing, and that is called as a plaque. So the plaque inhibition assay tells us how much of the plaque were prevented from forming or how much did not end up happening after a particular period of time. So the incubation period is about 4 to 5 days. And then we check how many plaques are formed. We count the number of plaques. The higher the number of plaques, the better the inhibition. And what you see is the inhibition of plaque formation in terms of percentage for each of the 7 molecules here, with the inhibitor concentration, which is diluted from 200 micromolar to 1.63 micromolar. And one particular compound showed a very good effect at about 3 micromolar. So now we have a molecule that is an initial uh, starting molecule, you can say, that can bind to the envelope protein and makes some interactions with the envelope protein. So this we ended up publishing in 2009. And what you see here is that validation part of the docked complex, showing which residues it's making an interaction with. And comparing with your own molecules is not enough. You have to compare with existing antivirals if there are. But since this was the first fusion inhibitor that was designed, we ended up did not having any comparison, but future work did show, and I'll show you that. Now, the next part, now this particular molecule which had bound with a 3 micromolar is what we call as a lead molecule. A lead molecule is something that you can use to make more other drug molecules. So the other molecules, since as I said, this molecule is commercially available, we cannot patent it. So to patent it, you need to uh, create new diverse molecules that are different from the parent or the lead molecule. There is a question in the chat box. The virus shows pH sensitive function. Are there pH related assays to validate? Probably there are, but then they are very difficult to measure. So, and also pH is very short. 1 second, just hold on. So the pH, if you drop it, it acts like it is not like a gradient. Immediately it will spread and it will lower or change the pH in the entire environment. And that is a very fast change. So those type of assays are difficult to control. So as of now, I don't know any assays. Even if it's that, it may be difficult to control the pH change. Okay. So what we did with this lead molecule, why do we need to change it? One, we need to patent it if we have to come up with a novel structure. The second thing is that the molecules IC50, that based on the dose response curve that you see here, it is about 3 micromolar. Now, any drug that needs to be identified or that needs to be marketed, you need it in the nanomolar range. If it is micromolar, then that means you need more quantities of the drug to be ingested or delivered through whatever route, administration in intramuscular, gastrointestinal, or whatever it is. So if it is in nanomolar, then the potency of the drug is high, and at the same time, you only need a very small amount of the drug to achieve the same effect. So when we look at the R1 molecule, which showed 3 micromolar IC50, we identified 5 fragments, and these 5 fragments are the ones, or we can say that we break it into 5 regions and we change one region at a time, keeping rest of them stationary. Like for example, if we remove this chlorohexane part with something else, the remaining fragments will be the same. So there is a tool where you can specify which part to change, and it will give you variations of the same molecule, but only one part change. So like that, we created a library. So that library of molecules were then again talked, and then we identified if any of these molecules are better binders than the existing ones. And we came up with a list, and we proposed that those molecules can be tested. Unfortunately, the molecules that we came up with this particular part is all hypothetical. they are present on the computer. You cannot change that in the or you cannot ask a company to synthesize. Even if they do, it will be very expensive. In medicinal chemist angle, it may be time consuming and the yield will be very less. So we stopped only with the proposal of the molecule or what molecules can be identified and what can be done. But this is way old work. So now there might be much better uh, ways of doing it, of testing, the high throughput testing in the lab, which will make life easier and also synthesis easy in it. Now, remember, there was another cavity which was predicted, and that means locking the conformation in the dimer state. So, this particular first cavity that we had identified an active molecule or a lead that has some potential is in the second cavity or the first cavity, and that is present between domain 2 and 3. So for the second cavity, again, we went ahead with getting a much larger data set, and then we talked only with gold because by this time we realized that the scores of gold that we get is then the ranking or the sorting of the highly ranked uh, molecules in gold and in X score were almost comparable. So we just went ahead with gold and then we identified 6 potential molecules which can be tested to try against the dengue protein. Okay. So again, we ended up looking at the interactions. Now, in this particular case, since it is present, it's locking the dimer. It has to interact with both the chains, A chain and B chain. So the residue should be interacting, should be having both A chain and B chain. And again, the gold score, you can instantly see the gold scores are far better. And the X score energies are also really good. So we ended up proposing these molecules, and we don't know who is taking care of it now. But it was a proposal that was proposed. Now, one of the things that as we have been seeing is we have been looking at the conformation of the envelope protein in the dengue virus in comparison with other flavivirus like West Nile virus or tick-borne encephalitis or Japanese encephalitis virus. So how do these molecules for the first site are binding with those particular envelope proteins? Why do you want to do this? Because there might be a chance that you are infused or infected with more than one virus. And if you are, then the drug that you're taking will be effective only for. So that is the whole reason why we wanted to see if we are able to find the very similar reaction between the two molecules and the two proteins or multiple proteins belonging to the same family. Okay. Similarly, the other site, so this is for potential molecule for site 1 in other plant viral, and the other plant viral proteins in site 1. So what you see here is the final image of the dengue viral envelope protein shown here is kind of representation. And can someone tell me what representation is this blue, red, and yellow of the protein?
Domain 1, 2, 3 regions, sir.
No, representation. We have different representation. One is file, Sitina, then uh, stick, then what are the representation?
Big.
Huh?
Hydrophobic.
Okay, then
Mm,
What representation is this? Anyone? This is the surface representation. Okay. So this is the cavity R1, and in that R1 is bound with, and you can see that the differences between the various flavor viruses envelope protein comparison with dengue. So there are a lot of variations in which it is binding to other flavor viruses. This is good and at the same time not so good. Why is it good? Because it has some specificity at this particular part and thereby making all the molecules bind to this with the golden how the surface representation. But we have diversity in terms of the later part of the molecule. That is because the cavity size and the residues that are interacting with these molecules are completely different. Coming to the next one where we did the molecular dynamics. Now, in this particular molecular dynamics, we again just did not take the structure and did the dynamics. We had a mechanistic hypothesis. What is the mechanistic hypothesis? Now, the closest one that uh, is known to undergo a pH-based conformational change is in the influenza virus. This is the hemagglutinin of influenza virus. At the low pH, what happens is that this blue cyan region flips and forms an interaction with other residues. And what you see is the histidine residues that are highlighted in this, which are seen as proposed as responsible for this low pH change. So that gave an idea that possibly in dengue virus also in the endosome it might be possible that the hydrogen bonds are making a role in the histidine residues where the low pH triggers the histidine residues to form another conformation. What atomistic detail we know from influenza virus is that you can see these residues. I don't know how clear you see it, maybe here. You can see clear. The histidine 184 makes an interaction with arginine 220. Similarly, lysine 51 makes an interaction with histidine 106. Similarly, this arginine makes an interaction with this histidine, and this histidine makes an interaction with this lysine. So in this particular case, the histidine, even though it has two amide group, and it is classified as a positively charged residue, it actually is classified as a aromatic residue. And in this particular instance, we are considering only the first part, that it is a charged residue. In at low pH, in the endosome, this influenza hemagglutinin undergoes a conformational change, and this conformational change makes the bonds between the histidine and the other residues disrupt. So that's what you see here. The histidine makes some other bonds. So similarly, the lysine histidine in the dengue viral, in the dimer, what happens is it dissociates the existing bond and makes another bond in the trimer. Okay. So for example, here this histidine is bound with arginine 9. In the trimer, this histidine is way far away from arginine. It can't make any more of those bonds. So, what is the hypothesis? The hypothesis is that this is the region where the histidine, the imidazole group of the histidine is bound. When low pH happens, this histidine is repelled out of its cavity because the two amide bonds are protonated at a low pH, and this protonation makes the ring, imidazole ring, to push out of its existing place, and it flips to bind to another negatively charged amino acid. So when we looked at, irrespective of how many histidines are there, if we protate all the histidines and we run a simulation, we see these small gaps. That means at some point of time, the dimer is dissociating. So the hypothesis was that if all histidines are required, what particular histidines are conserved? One second. One second. Sri Mary Lena Aruldas. Sorry, there is some inquiry happening and I just need to attend that. Meanwhile, I'll look at the question. So the question is, dengue virus has RNA as genetic material. Are there any chance of high mutation rate in viral attachment? Yes, that's a good question. So compared to other RNA viruses, the mutation rate or the rate of mutation of the amino acid changing, that is the RNA mutation changing and giving rise to another amino acid, it's very slow compared to other RNA viruses. So it's more like a very slow virus, and the evolution doesn't affect the mutation. So that much. So that means if we use a drug and we focus on a residue binding, that residue will not change over a period of time. One second. Okay, the next question is, which is more efficient antiviral target? That depends. For us, we at that point of time, viral entry, we wanted to inhibit and design fusion inhibitors, and that was we thought was more efficient. But there could be some other more efficient based on more recent reports and more recent experimental data for dengue viral proteins. So we know that all histidines, if you rotate the structure, can undergo some kind of, you can see the dissociation happening at some point of time. This was 20 nanoseconds. So, but then at that point of time, 20 years ago, computational power was very limited, and we wanted to focus on the simplest scenario on first principles. So we said, okay, why not we look at the histidines that are completely conserved, that is 100%, and is present in a hydrophobic pocket. Because if you look at this, it has to be present in a hydrophobic pocket to make this interaction and then expelled out. If this histidine is present on the surface, whatever histidine that is 100% conserved, that will be amino acid on these four histidines. Are these histidines acting as a cooperative act to trigger the change? And whether this histidine acts like a link pin. So what is a link pin? Link pin is nothing but you might have seen in a real life analogy, something that is holding the entire structure together. Okay. So if you remove that particular component, the entire structure collapses. So that is called as a link pin. You can say the weakest link. Maybe the histidines are the weakest links. And it is probably the low pH triggering the conformational change. We don't know. So these are the questions we went about. So we started with two histidines, 144 and 370. We had four separate cases. The first case was that where we mutate the histidines to alanine, and the protonation, the side chain absence and protonation is giving rise to a conformational change. In the second case, we do the mutation, but we do not protonate, which means that the side chain, if there is a conformational change, then the side chain is giving is responsible and protonation is not playing a role. In the third case, we do not mutate, but we protonate the rest of the histidines. These two histidines are not protonated, which means protonating these two histidines is enough to in this particular case, only two histidines are protonated, not the others, which means just these two are enough to undergo the conformational change. Sorry, I said the other way. In this particular case, H144H, in this other histidines are protonated. These two are not protonated, which means histidine protonation is a necessary condition for conformational change. And in the case 4 is what I had mentioned now. Only these two are protonated. So this is a sufficient condition for conformational change. So a necessary conformation and a sufficient condition. So we ran about 20 nanoseconds at that point of time. That was the computational resource we had. When we look at the root mean square deviation, RMSD, of the four different cases, black, red, green, and blue for case 1 to case 4. X-axis is the picosecond time. So 20,000 picoseconds is 20 nanosecond. Here it is in nanometer, which means this is 20 angstroms and this is 30 angstroms. 1 nanometer is 10 angstroms. This is 0.2 angstrom, 0.2 nanometer. So this is 20 angstroms. So we see that there is some changes happening globally. We also looked at radius of gyration. Radius of gyration is, one second, I'm getting a cough here. Excuse me. So radius of gyration tells us if the molecule is undergoing a conformational change in terms of its overall structure. Again, a global property. This protein, as you might have seen by now, is very linear in nature. It's like a rectangular or a cuboidal shape. So the protein structure needs to remain in that particular shape for at least some period of time. What radius of gyration tells us is if the protein becomes globular or the protein becomes highly unstable and it unwinds itself. And when it does that, the radius of gyration will increase. But we see in all the four cases, the radius of gyration is not increasing so much. There is fluctuation, but we don't see many global changes. So it's a very stable setup. And then when we looked at all the bond information of which particular arginine, which particular glutamic acid are making ionic bonds, and due to the protonation, they are getting broken. In case 1, case 2, we see these bonds are not broken. Black and red, they are almost present one over the above. But in the case of the mutant, in case 3 and case 4, where we protonated all the histamines, protonated only those two histamines, we see those ionic bonds being broken at some point of time. Here, around 15 nanoseconds, it gets broken. Here, at the very beginning itself, gets broken and remains broken for a long period of time. When we look at the region where this particular fusion peptide is present and the mutation, we see some root mean square fluctuations in that particular region, which means those residues are playing a role and they are also getting affected because of this. So the conclusion was the protonation of these two histamines was sufficient enough to cause a disruption of the salt bridge in chain B. And the 20 nanosecond simulation told us good enough data that the dimer is in a metastable structure, meaning that it is just waiting for the protonation or to be in a low pH environment and it starts undergoing a conformational change. Then we thought, okay, Campman et al., which is also my colleague, Thorsten, what he did was he did with triple mutant. So he showed three histidine residues are 100% conserved. We also saw four histidine residues are 100% conserved. So do we want to try this with three mutants and three histidines? So we ended up doing that. So again, four different cases, having a necessary condition and a sufficient condition. And again, we see here, it is not very similar to the other, where we see some quite a good global change happening in the RMST for some of the cases, especially for the first case, where the histidine has been mutated to alanine. And you can see the blue, that is the case for the RMST increases at the end of the simulation. So the radius of gyration is not the same. It shows that the molecule is bending. You see that it is bending, which means it's curving, which means there is a hinge motion that is happening, because of which the dimer is trying to bend itself. Okay, maybe it is bending and then releasing it out into the trimer. When we look at the salt bridge information is very stable. And then, but then we look at the root mean square fluctuation of those residues in the fusion peptide, they are highly fluctuating. So comparing the structures at 0 and 20 nanosecond, they show that these three histidines are also playing a role in the structure, but exactly how it's playing a role, it's not very clear at this point of time with the salt bridge, with the root mean square deviation and the root mean square fluctuation and the radius of gyration. Some of them are global parameters, some of them are local parameters. But how much is enough? Should we go ahead with four mutants? So we can keep on doing that, but it will only add more to the noise. There might be some insights obtained, but then there are some long range interactions happening at these positions, because of which there is some conformational change being happened. So what we found was, even though some changes are observed, given the time scales that we could do, we don't know whether a specific residue is absolutely necessary, but we know that at least two or three residues are primarily responsible for inducing the initial stages of activation. Maybe if we continue the simulation for a longer period of time, we might end up having a much better mechanistic. So this work was done at JNU with Professor Naidu Subbarao, Professor Alok Bhattacharya, and Professor M.C. Samanthi. And this was a collaborative work with University of Queensland in Australia with Boschard, Paul, Alan, and Ross. And with that, I'll take any questions that you have.
Sir, the inhibitors which we have to identify our target is based on uh, post-fusion virus entry, sir.
No, we looked at both pre and post fusion, and we looked at cavities that are present in the pre-fusion but not in the post-fusion. So we targeted the dimer in the virtual state.
So glycosylation is only the difference between pre-fusion and post-fusion, sir?
No, there is a conformational change. That is what we focused on.
Okay.
Because the virus enters the host cell, and when it enters and when it exits the host cell, at the end as a mature virus, glycosylation happens. It's a natural process. We cannot prevent glycosylation, but we know which residues are glycosylated, and we also can see glycosylation affecting that particular cavity in binding to a particular inhibitor or not, making the site available to a particular uh, inhibitor. Any other questions?
Yes, participants, you can ask question. I am also unwell today.